Hi everyone, I'd like to introduce our lab, the Gácser laboratory, where we are working mainly about the host pathogen interactions. So we investigate both the pathogen side as well as the host side. The major question that we are asking in the pathogen side, what makes a fungus pathogen? We are investigating the fungal valence factors. On the other side, how the nature of the host response is orchestrated during the infection, and we investigate both the innate and humoral immune response. So why we are working with pathogen fungi, mainly with Candida species? They are causing a very severe health problem, causing more than 50,000 infections annually, which can be invasive, and if it's invasive, then the mortality rate can be as high as 46 or 70%. So maybe you heard about the Candida species, especially the Candida albicans, or nowadays the Candida auris, which is causing very severe problems, especially in the United States. And these are all Candida that can cause disease in humans. So in the case of the pathogen side, we are interested how they are able to survive in our body. So for example, the nutrient acquisition is one of the major characteristics of these pathogens. And once we are trying to combat them, then we have to use drugs, antifungal drugs. And unfortunately, some of them can be develop drug resistance, especially Candida auris is very good in it. On the other side, we are also working with the epithelial barriers, the host epithelial barriers, and also how our immune cells are interacting with the pathogens. And for that, we use different models, such as cell lines. As you can see here, these are macrophages, murine macrophages from the mouse or human macrophages, but we also use in vivo systems. These are the conventional ones, like the mouse. But we also use non-conventional models such as Galleria melonella larvae. For the method repertoire that we are using is quite large. We are using immunological methods such as ELISA, or we have the imaging facility which is focusing on the capturing the cells. On the host side, we are investigating the communication between different cell types via extracellular vesicles. We use a lot of in silico analysis, RNA-seq and this kind of methods. Our recent focus is actually investigation of the tumor pathogen interactions. So the major question is how the pathogen such as Candida albicans triggers oral squamous cell carcinoma. So this question is actually how a cancer, which is a completely different disease than an infectious disease, interacts with the oral squamous cell carcinoma. And we also investigate what is the trigger during OSCC progression. For that, we investigate how the surface of the fungus cell can interact with the oral squamous cell carcinoma. We also investigate the vesicles that are secreted both from the tumor and the fungus. And we then analyze all the molecular mechanisms that are in link with cancer development. We are also interested in different other areas, for example, vaccine development against Candida species, which can be revolutionize the combat against this kind of pathogens. If you are interested in our work, then you can also watch the homepage of the laboratory. So I finished my PhD at the University of Szeged, and I received the Marie Curie Fellowship, and I started my first postdoc in Hamburg. the University of Hamburg, where I actually started to work with fungal pathogens. And then I had the fortune to work in the United States, in New York, at the Albert Einstein College of Medicine, where I worked with Josna Sanchuk, and with his lab we were actually focusing also different fungal pathogens such as Candida and other important fungus like Cryptococcus, which was the main cause of death in the AIDS pandemic. So I am a university professor working at the Institute of Biology, and since 2018 I am also the head of the institute. This is a quite large institute. We have 10 departments, including immunology, molecular biology, physiology, and so on. So all areas of the biology is covered. And my lab is mainly focusing on microbiology and immunology focused research. And I am actually affiliated at the microbiology department. Infectious diseases caused by fungal pathogens are quite large health problems. So especially because they are really difficult to handle. I mean, they are eukaryotes. So in contrast to bacteria, which are prokaryotes, there are very few targets that we can use to combat against them so that we don't harm the host. So that's why it's a very challenging area of research, and I think there are very interesting new areas developing recently where we are trying to show that these infections can also interfere with our health in general. For example, if you are getting any type of cancer, especially in the oral cavity or the gut, then can be influenced with this type of infections. And more evidence is showing that that is an important field of research. So I would like to encourage you to stay on this field because science is always really interesting and very diverse. So you will never feel that you see always the same faces and always the same problems. And you can meet very inspiring persons like we have the fortunate to work with Katalin Karikó recently and potentially developing vaccines against Candida infections. So it's a very diverse environment and a very inspiring environment at the end. But I have also mentioned that this is a very hard work.